Yes, ma'am. Yes, ma'am. Yes, ma'am. No problem. Ma'am, in title.
Good afternoon, participants, and good afternoon, sir. I am very glad to introduce the speaker of this session, Dr. Ramakrishna, sir. He has done his doctorate in IIT Chennai, IITM Chennai, and he has done an excellent work in his PhD. His work has gone to patent level. I think it has been granted also. To his credit, he has got a lot of publications in reputed journal, and still he is carrying on, he's continuing his research. Currently, he is working in SRM Institute of Technology and in EC, Electronics and Communication Department. If you want to do some projects in the area which he is going to discuss today, you can come to Instrumentation Department of SRM Institute and you can do the project under his guidance also. You're most welcome to spend one month or 2 months in Instrumentation Department, and you can associate with him for the research ideas and bring out any product, or you can carry on with your research work also. Now, with these few words, I hand over the session to Dr. Ramakrishna, sir.
Good afternoon, everyone. Good afternoon. I hope that you can hear me. Is my voice clear? Is my voice clear, ma'am?
Yes, sir. Yes, sir.
Okay. Thank you. Thank you very much.
It's louder.
Yeah. It's a little louder. Huh? Okay. I'll reduce the volume. Is my voice clear now? Is my voice clear now?
Participants, if you don't hear his voice, please put it in the chat box.
Okay. Okay. Right. Thanks for this afternoon. Welcome to give me a lecture here on on-chip smart materials, micro devices, fabrication techniques, and characterization for emerging applications. Okay, I will start from one of each and every word. That emerging applications. Yes, of course, this talk is actually well suited for this emerging applications only, and this topic is more suited for lab-on-chip smart materials kind of and micro devices. So I'm not talking about the fabrication and characterization here, but I'm mostly intended to discuss about lab-on-chip smart material, what kind of materials that we are using, and what are the applications that we are targeting here. I'm going to discuss in this talk. So actually, initially in the brochure, the topic was smart materials. Okay, okay, just a minute. I'll go through my PPT and then I'll put it up. Okay, smart sensor modules for biomedical application is a broad topic that has been mentioned there. However, we will be talking a very focused topic, that is fiber optic sensors for biomolecule detection. So especially I'm going to talk about fiber optic sensors and how are we going to detect the biomolecules in general. That is what the topic today. So I'll share my screen with you and then I'll continue the talk for the next 2 hours. So just give me a moment. I have to share my screen. Okay. I hope it is visible for you. I hope it is visible for you.
Yes, sir. Yes, it is visible.
Yes, ma'am. Yes, ma'am. Yes, ma'am. Yes, ma'am. I'm doing it. Now it is full screen, ma'am.
Yes, sir. Yes, sir. Scroll it, sir. Let me check.
Yeah. I hope everybody is able to see this full screen mode. So this is the first slide. All symbols are there, who are actually organizing members and organizing committees are there involving. So today's topic is focused on fiber optic sensors for biomolecule application, which is actually come under sensors module for biomedical applications only. So let me take this content too, because people should know what is that I'm going to talk for the next 2 hours. So the first thing that I'm going to talk about the motivation and brief introduction about what is that research that we are actually progressing towards and why am I actually doing this particular research. That I will give you a small motivation. Of course, I will draw you some objectives also. What kind of work that we need to take up. That objective 1 I will be discussing here, and later I'll be discussing about some biomolecular detection conventional methods are there. What are the and what are the limitations over there, and why we need to go for smart sensors, and further discussions will be there here, of course. But before going to discuss the smart materials and smart sensors, we need to know and we have to have some idea about the existing technology and what are the limitations over there. Of course, there are very good positive points are there for existing technologies, and hopefully we are actually using them right now in market or even in the clinical diagnostic industry. However, there are certain limitations that poses some research focus towards the smart materials and smart technologies, because everywhere we wanted to carry them. So how we are going to design, or how we are going to develop the second objective, that smart materials, how we are going to go for that we are going to discuss here. And the next topic that I'm going to discuss is fiber optic sensors and its types and applications. What are the types of fiber optic sensors that are available, and what are the applications, in general applications we are going to discuss. And then later we'll going to going in detail that intrinsic and extrinsic fiber optic sensors. That is the focused topic that we are going to discuss, and how they become a biosensors, that is also we're going to discuss. And then plasmonic particles. Of course, nowadays everything into nanomaterials and nanotechnology is coming up, so nanomaterials also we'll discuss here. And that participation, the conjunction of nanomaterials and for fiber optic sensor, how they are going to give you some benefits in sensing modalities that we are going to discuss. And then I will give you some recent development of this fiber optic sensors from our research point I will discuss with you. So this is a brief content tour. So people might have understood this is what I'm going to talk for the next 145 minutes and all. So please bear me here I'll start with some motivation here. However, we are all in the COVID pandemic situation. How many of you really know about whether we got COVID or not? So, but basically what we do is we are only looking at the symptoms, whether you have cold or cough and temperature rising. These are the symptoms are actually giving you an indication of that, okay, there may be a COVID, but we are not sure whether it is COVID or not. It may be a common cold or common temperature also. See, most of these diagnostics at your level, maybe it is with symptoms. Even if you consult a doctor, if you go to a clinical clinician or doctor, what he will do actually in the initial stage, they'll ask for the symptoms. However, they will not confirm you the medication, because symptoms will never give you the whole idea of what exactly the disease is, because symptoms are the natural signals. The symptoms are the natural signals to recognize there is a disease, but you cannot exactly identify this is the disease. So we need some diagnosis to be done, which means that some chemical analysis or some biomedical analysis, something should be there to confirm this is the disease. So some specific test we need to undergo, and then we will confirm, yes, this is COVID, this is malaria, or this is some other disease. So to confirm, we need a diagnostic procedure. So no doctor will give you direct medication for any disease with only symptoms. So we need to confirm that particular disease by diagnosis procedure only. So now you see that if you are looking at this particular scenario, 70% of medical decisions, when I say medical decisions, means I wanted to give you medication. For example, this patient requires a medication. So if you wanted to take that decision, you need to rely only on the clinical diagnosis outputs. The diagnostics outputs only will give you this particular, give you a chance to decide what kind of medication you need to take. So other 30%, you may say common cold, common temperature and all, these people will give you some paracetamol, and these are the small advisory things. But however, for the major diseases like malaria, dengue, and COVID kind of diseases, where you think you need to do a proper diagnosis and then confirm the medication for the patient. So now at this topic is more relevant to us, because we are discussing about immunosensor, which actually recognize the antibodies of COVID antibodies that has been produced in your body, or maybe malarial antibodies or dengue antibodies. So it's like, whatever that immune system response that is coming, and those molecules that are present in your body can be identified. So how we are going to do that, we are going to discuss in this entire due course of next 1 minutes. So now you see that before going to do any diagnostic tool, for example, we have just discussed about COVID, malaria and other things, but look at our country in general. So if you look at that, our is the second largest population in the world. Please try to understand this is very important topics. I can say it's the facts about India. It is, I mean, the numbers maybe here and there, the plus or minus will be there. However, but these are very, very close facts. So the second largest population, which means almost like 130 million people are there, and then out of them almost like 67 to 70% people are actually living in rural area. They are not into metropolitan cities or even second towns and all. They are actually living in rural area, which means like less than a town, a village to village, or semi slightly developed town kind of. And the another fact is 22% of people are actually under the poverty. Poverty means, when I'm talking about financial status, they are unable to even even buy a small paracetamol available outside, because they cannot spend much money about it. So if you look at these people, if you ask a diagnostic tool, go and get a report, I don't think so they will actually do that if the test is too costly. So now, how can we look these facts? When you're actually looking at smart materials, smart sensors and all, you wanted to do something smart way, what is more important is where you're going to utilize them. Country like India, you have to understand the burden, the disease burden, or the human population. All these parameters are actually important to decide what kind of diagnostic tool that you wanted to develop and all. However, next come to the important point is mortality rate, and in that we have two things. The one is communicable diseases, and the second one is the non-communicable diseases. Please try to understand this is very, very, very important. If you are looking at a smart sensor design person, then you have to understand these two parameters very, because you are going to draw a major objective from these entire facts. So what is it mortality rate showing? Almost like 35% of mortality rate, I mean, upon all the deaths, 35% or maybe equal to 35%, people are actually dying because of TB, HIV, diarrhea, dengue, chikungunya, and malaria. Please try to understand, at these situations, when diarrhea or dengue or chikungunya is spreading, Deepan Chakravarti, please mute your mic. Okay. Dengue and chikungunya and malaria, these are the fast spreading diseases. When a disease is spreading faster, then we have to have a diagnostic tool which should actually mass screenable, because if I have a diagnostic tool and it gives you output at almost like 1 hour or 2 hours, it may not be useful, because within 1 hour almost like 100 and 200 people are coming to the hospital and you cannot actually survive. So we need to have a sensor could be mass producible and should be applicable to everyone, immediately we can do. Should not have a single module like that. So the device should be a mass screenable kind of. And another thing is non-communicable diseases, because it is very important. Non-communicable diseases, mortality rate is 5. For example, cancer, heart attack, and other causes of death and all, that comes under non-communicable. However, these diseases can be identified much early, which means that the onset of the disease can be identified much early, so that we can actually give the medication and reduce the mortality rate. So in this situation, the device development, the smart sensor or whatever that you are looking at, the materials, sensing modality should be much more sensitive to the sensing environment. For example, if you're going to sense a chemical molecule or the biomolecule or any other parameter, it has to sense much lower quantities. So this is very important. So from these things, I am going to give you the a large objective for the design and development people. So when I'm saying there's second largest population, which means that there is a huge potential for healthcare developments. For example, if you wanted to develop a diagnostic tool or maybe another modality, maybe pharma industry, whatever, the healthcare department is having the huge potential. Now you can actually work on. We have a largest population, so the market is good. And the second one is the off-bench diagnostics. Why we want to do this? Because most of the people are actually living in rural area. They can't travel to the metropolitan cities to get this particular diagnostic done. So if you wanted to move this diagnostic tool to the rural places, it need to be an off-bench diagnostic tool. It should not be a fixed, like it's like a big room. You cannot move it. It's supposed to be a mobile, and you have to take it. So there we are going to look for lab-on-chip modalities, or maybe off-bench modalities kind of. This next one is that affordability also more important, because we are targeting the more people like living lower than your poverty line. So there also we have to look for the diagnostic procedure or the tool development should not go should not be a higher cost. So, and the other one is, I already discussed, mass screening capabilities and sensitive to detection. These 5 things are very important for a development person, because we have to, this is the objective number 1. I'm going to tell you, this is the objective number 1, is that design and development of a portable, low cost, efficient and easy to use diagnostic tool for the clinical diagnosis. This is very important. This is the objective 1. But this objective is the layman objective, because a layman, of course, it's a common objective for everyone, because it is like, these are the facts we are going to target it. But the second objective is much more important. What kind of sensor that you want to design, how you are going to design, and what kind of physics phenomena or chemistry phenomena that you're going to take up. And this is very important, that we will discuss in the next slides. So the first thing that we wanted to tell you is we are looking at biomolecule detection. When I'm saying biomolecule detection, why am I talking about biomolecule detection? Here, when we are actually going to any clinical diagnostic procedure, they will collect the serum or your saliva or maybe the urine samples or the blood samples, and they are actually looking for the biomolecular presence, whether the molecules are present or not. So, or maybe some protein content, whether is it for present or not, or maybe some DNA content is present or not. So these are the things that we are actually looking at. Not talking about other procedure like MRI, ECG and all. Those are also biomedical tools. However, that diagnostic is completely different. But here, when you come to the biochemical analysis, here we are actually looking at the content, like DNA content, or ribosome content, or maybe lipids content, or maybe proteins content. How much it is, how much is available, or whatever the targeted molecule. These are the things that are more important for us. They are very specific the diseases, so we can identify disease at very precisely. So for example, here I'm giving the biomolecular list, mostly like lipids, nucleic acids, carbohydrates and proteins. However, other biomolecules are also there. For example, primary and secondary metabolites, some carbon based materials, including glucose also. You can actually include glucose also into that. So why am I saying glucose? Because glucose sensors are already available in your market for diabetic analysis and all. So that also we are going to discuss in next slides. So when I say biomolecules, most important thing is nucleic acids and the proteins. You can call it as protein or the immunoglobulins, because it's a globular form of protein chain. It's like a structure. It's a immune structure. So where it is very specific to the particular diseases and all, or this is antigens. You can call it as antigens. So now you see that what are the conventional analytical methods that are available right now. If I need to do some blood analysis or urine analysis or maybe the saliva analysis, what kind of techniques that we have? We have conventionally two major techniques, that is ELISA and the PCR techniques are there. Very good techniques, very efficient ones. They will actually give you immediate data. However, for them is a little taking a lot of time, because it cannot be immediately useful for mass cleanability or immediately sensing for the very low concentrations and all. So there is some limitations here, but however, these are the two important conventional techniques which will give you a good idea or the basic idea about what kind of molecules are present inside. So now you look at the ELISA. Here it is purely based on enzymatic reactions, and the second one is the antigen antibody specific reactions. Both are actually happening here. The antigen antibody reaction, that immunoreactions, are actually measured in this case because we wanted to trap the molecule of the interest which is given in the sample, for example blood or urine or saliva. So that molecule will be captured. After that we will be having an enzyme over there, and this enzyme will actually changing the products into substrates into products. So these products will actually give you some color output. So by looking at the color, we will confirm the presence of the interested molecule, which is trapped, which is actually trapped in between the two immune molecules, two immunoglobulin molecules. So confirm, okay, this molecule is present. And of course, the color, the texture of the color or the intensity of the color is depending on how many particles are actually trapped. It depends on, this is a number game only. If number of particles trapped is more, number of ERP, that is Harshad is enzyme is more, then the color turning color will be more. So it is like a qualitatively we can actually study by looking at the color. After that, people have tested, there are down concentration down, and you can actually find out what is the concentration in that by just doing the analytical fit and all. However, this requires a minimal amount of targeted protein to be trapped. It should be trapped. Otherwise, you cannot actually detect the color. This technology is very good if you are actually looking at naked eye detection, you can see the color change with your naked eye. Of course, there are some modalities that came to read the color using some scanners and all that are also there. The detection limits are actually come down. Next technology is the PCR technology. This is purely on DNA based. What will happen if you are taking a sample, if the DNA molecule is present, and that DNA will be amplified for 40 to 50 times, then it will go for the recognition event. So this is what in PCR happening. In COVID times, the PCR test is actually done well. However, now we are looking at, see, if these two technology, if you wanted to adopt, you have to spend a lot of time you have to wait a lot of time to get the results done. So that is why we are actually looking for smart technologies, which will give you immediately the moment you give the sample. I wanted to know whether COVID is present or not, or maybe the particular disease is present or not, or maybe the person is expected to have a heart attack or not. These are the things that we have to identify immediately. So let's look at each and technology. Eliza Technology, you see that there is a capture plate, where the microplates are there, where some capture antibody need to be coated. It takes at least 12 hours to 18 hours to coating itself, and after that you have to add the sample and then wash it, and then again add another recognizing antibodies which has the enzymes, and after that you have to wash again you have to put the substrate. Then there will be some time it will take to react and give the results, and after that you are going to reading. So there are so many steps that are involved, like 1 to 8 steps are involved in that. Then of course each step requires some required amount of time. So you see that ELISA, if you wanted to perform, it takes little time. Of course there are 4 types of ELISAs are there. I'm not going in detail into the different types of ELISAs. And after that, the second technology, very important one, is the polymerase chain reaction. What is the PCR? Here what is the important thing is, the first step is we have to denature the DNA molecule. You have double strand, you have to denature them. Again, you have to coat them. Like that, you have to do that amplification for 30 times to 40 times. See, these 3 processes, for example, one is the denaturation and annealing and elongation. So this process has to be repeated for 40 to 50 times to amplify the DNA concentration. So then only you can go for recognition event. This is very important for example, if you go for your COVID test, what is that they're going to do? They're going to take the swab from your nostril, and then they're going to put it in the lysis buffer. Then the DNA need to be amplified much more, and then identify what kind of DNA is present there. So it is also taking a lot of time. So it is better to go, we're actually looking for other technology which will give you immediate results with very consuming very less times. So this is a very familiar technology, might be everybody knowing this. This is the lateral flow or dip type dipstick technology. What it is actually, if you just take a sample, a blood sample or the urine sample. Excuse me, somebody is speaking. Srinivasan. Srinivasan sir, please mute your mic. Srinivasan sir, please mute your mic. Thank you. Okay. So now you see that lateral flow technology, where you have simple beds are there, bed pads are there, where you are going to give your sample input. This sample input having your analysis molecules are there, and these molecules will be traveled through the conjugated bed. There the nanoparticles are there. Of course, the pooling of nanoparticles will give you a turnout some color. Could be a black or pink color, it will come, and you get to see some pregnancy test, otherwise a cancer test and glucose test. People are actually doing with lines. You just put your urine sample over there, and within 15 minutes or 12 minutes, you get to see the colors are happening. The lines are appearing to you. If the 2 lines are appearing, it's a positive sample. So this is a very good technology. People have been looking everywhere. It is available, even in the market is available. However, the readability of this particular technology, for example, how much concentration it is a qualitative analysis. For example, glucose or the pregnancy confirmation, or maybe heart attack other molecular confirmation can be done through this particular technology. However, the qualitative is there, but quantitative is not possible by naked eye. So for this also, the strip readers have come. People have come up with the strip readers, and of course they are going to read that particular 2 lines intensity and all, and then they are actually giving some concentration things. But we want to know the much better technology than this, because it is also taking same time and all. But the reading, if you wanted to read this molecule, you need to have have higher concentration, because that line need to be appeared even for the detection systems. The line need to be a little thicker so that it will be identifiable. So these technologies are right now available. But compared to these technologies, there are other technologies also came into market which are much more sensitive. Very, it's like automolar. If you say that only 100 molecules are present in one 100 microliters, in that cases also, if you wanted to detect, so these are the technologies that are going to help you to capture the molecules in the 100 microliters wherever it is, and then trap them into the wells, and you can identify them. It is like this technology is called Simoa technology, which is very famous right now, and the digital ELISA, you can call it as digital ELISA also. If the sample size is very small and you wanted to capture the molecule, then you wanted to use this microbead technology. These microbeads are actually coated with antibodies, and these antibodies are actually capturing the analyte molecule which you wanted to find in the liquid phase itself. Once it is captured, these beads will be trapped into the wells. These beads will be trapped into the wells, and then you are going to give the substrates, and then the color change will be happening. However, the color change will not be visible to you because it's a very small chip. It's a very small chip. Only each well actually combine only 25 microliters, or very less than 20 microliters or 10 microliters sample, only very less sample you are going to use there. Once these beads are trapped, and this particular wells will turn out into colors, and we are going to find out how many wells are actually turned out into colors. Will give you the number of analyte molecules over there. So this is the technology, very famous. However, this also required some addition of substrates and changing in color. Again, you need to have a readers over there. The scanners and readers are required. The electronic items are there. And the next technology people have tried with the biobarcodes. It is just like an amplification technology, like your PCR. In PCR, what you have done, one DNA you have taken and multiple copies you have generated. But here in this technology, one immune molecule will be trapped with two beads. One is a magnetic bead, and second one is a plasmonic bead, that is a gold bead, gold nanoparticle. And now you see that after this trapping, we are going to keep them down, because the magnetic particle, you just apply the magnetic field, they are all actually coming down, and the other liquid we are going to remove through it. And then again you remove these molecules, where the DNA molecules are actually amplified molecules. Number, for example, it is 1:10 or 1:100 or 1:1000 could be there. For example, one analyte molecule will give you n number of DNA molecules output, and then you can go for hybridization. Again, you're going for gold nanoparticle based sensing modalities. This is a biobarcode technology. Here also, this technology is very famous, and for automolar and femtopolar detection, it is very, very good. But this technology, you see that there are some procedure is there, and a lot of steps are also involved. But again, finally, we are going to use a reader technology. You need a reader technology to identify the color, and wanted to quantify the number of molecules that have been trapped, and all, which means the concentration that you trapped. So it requires also a reader. So another technology came into picture, that is SPR technology. This is surface plasma resonance technology for biomolecular detection. Here what happened, there is a gold layer, that is gold coated prism will be there, and you are going to shine the light. One particular wavelength of light will be trapped or absorbed, and this wavelength is sensitive. The absorption wavelength is sensitive to its nearby environmental refractive index change. So if any molecule that is coming close to the gold plated surface, then there will be a shift in the absorption wavelength. That people have tried using this technology for biomolecular detection also. However, if you wanted to amplify this nature of sensitivity, people have tried with the nanoparticles, so the gold nanoparticles also. There people have reported 1 automolar, 1 femtomolar, detection types for Alzheimer disease and cardiac markers. For Alzheimer disease, you see that tau 381 is a marker, and for example, cardiac markers, for example, BNP is a cardiac marker, and thrombin is there. So these are the markers that people have been identified. Now you see that in a nutshell, there are a lot of technologies that are available right now based on its physical and chemical principle. For example, SPR, SMR, NW. This is a surface plasma resonance, and LFA means lateral flow assays. MMR means magnetic resonance rings. Resonant rings are there, and QCM technology. QCM technology means quartz crystal microbalance technology. and BBA technology, biobarcodes. MC means micro cantilever technology. There are many technologies. Different modalities have come. Optical based, magnetic based. Cantilever is mechanical based. There are many scientific based technologies have come, but you see that all these technologies are actually above one particular line. You see that there is a line I have drawn here. I mean, not I. This is Orlet has given in 2011 that limit of detection versus analysis time is a very important factor. If you are a designer, if you wanted to design a good sensor, so you have to look for the limit of detection and the analysis time. If you do that, there is a gray area where people need to explore, where the people need to explore whatever the existing technology, it is above the line, which means that if you are looking at the limit of detection, we are compromising on analysis time. If you are looking at the low analysis time, we are compromising on the limit of detection. You see that all these technologies are actually given in different color, and that colors, that outputs are actually given here. You see that all are above the particular line. So what is our targeted research? For example, if you wanted to do a sensor modality, what you're actually targeting is, we wanted to target this gray area. We wanted to come down in terms of limit of detection and also the analysis time. We wanted to detect the femtomolar detection, but we don't want to spend too much of time, that is around less than 10 minutes or less than 1 minute. We wanted to try out this. So this gray area is more important for the researcher. So now the second objective is, we wanted to develop a sensor which have very good limit of detection, including the less analysis time. This is the second objective one has to draw from this figure. So if you are actually developing a biosensor, we should understand what is the biosensor and what is the normal sensor. The sensor is actually giving a readable output. Any sensor will actually give you a readable output in terms of any other energy form. But no, you cannot call every sensor as a biosensor. The biosensor has a specific name called bio. Why this bio has come? Because this event is happening only by the biomolecules. There are specific biological events or biological phenomena that we are going to use to identify. Srinivasan, please silence your mic. Now you see that the biosensors are actually given. Any sensor, if you take any thermal sensor or a mechanical sensor, or maybe optical sensor, you take any sensor, that sensor need to be coated with a biological recognizing element. It has to have a biological recognizing element, then only you can call it as a biosensor. So any analyte that you wanted to detect, for example, DNA molecule or immune molecule or any other chemical molecule you wanted to detect, if a bioreceptor is placed on a sensor, then that sensor is called as biosensor. So now you say that any transducer, you can take any transducer that need to have a biological coating where it actually recognize the biomolecules. And the other thing that we have is the analytes that comes and bind. So this binding event will give you some change. This binding event will give you some change either in terms of electrical properties or mechanical properties or optical properties can be identified. Okay, the next one here, most importantly, we will talk about the fiber optic sensors. They are actually comes under optical biosensors. We are not talking about other modalities like the other lab on chip technologies where we are talking about micro resonance rings and all that. We are not talking about here. We'll simply talking about fiber optic sensors, how they are actually important for us. Fiber optic sensor. When I say fiber optic cable, it is a very tiny cable. It just two concentric cylinders are there. It's a long cable. You can travel. You can send the light through this cable, guided light. So you see that if you launch light one place, you can actually collect light from the other place. So through this channel, the light is transported. However, the light transported inside the fiber is sensitive to the fiber characteristics. For example, fiber parameters. What is the size of the fiber? What kind of material of the fiber? And what is the environment around the fiber? All these things will influence the light traveling inside the fiber. So when we are actually observing this phenomena, when we sensed it, light is sensitive to its intrinsic properties and also the extrinsic properties, then why can't we use this as a sensor? Yes, we can use this as a sensor. People have developed different types of varieties of sensors, and that we are going to discuss now. First of all, before going to discuss about this fiber optic sensor modalities, let's look at its characteristics. Fiber is like, very portable and cost effective also. There are different materials, silica materials and plastic materials are also available. And now you see that fibers can actually capable of carrying almost like, ultraviolet, which is 180 nanometer onwards to almost like micro, mid IR, that is 10 micron. Of course, the special fibers are required. Visible region and near IR region also we can actually transport. The mid IR is actually need a special fibers. And now you see that the moment you have this light is going in all these different wavelengths, now we can actually do some spectral analysis. The spectral analysis will give you the details about the sample of interest, which is around or on or inside the fiber. So now you see that lab can be on fiber or the tip. Lab on tip, you can actually create any sensing event that is happening on the tip, or you can actually do some sensing which is inside the fiber, or you can actually do some sensing around the fiber. So if you take fiber, there are three modalities people have identified: lab on tip, lab in fiber, and lab around fiber. There are three different techniques people have used, and of course you see different technologies how they are actually different. Let's look at the right corner, lab on top of tip, which means that there are coatings at the tip, the polished tip. There will be some coatings, either the nanoparticle coatings or some biomolecular coating. Different modalities people have tried. This is actually sensitive. Here people are trying to do the reflection and transmission based analysis. If the light is passing to the tip and coming back, when some event is happening, some recognition event is happening at the tip, will be recognized as a reflectance spectroscopy or transmittance spectroscopy. We can see that. And the second one is the lab inside fiber. There are halo fibers and dual core halo fibers and other photonic crystals are available. If the molecules that are actually able to go inside the fiber, in the gaps of the fiber, and then they will actually cause some change in the light intensity patterns or the light frequency patterns. The spectroscopic analysis you can do. So these two are something I'm not going to discuss in the next slides. Lab around fiber is more important. That's where I have actually contributed something. So please look at this lab around fiber. As we already discussed, the light traveling inside the optical fiber is sensitive to the environmental changes. It's environment around fiber, which means that the change in optical characteristics of the environment will definitely affect the light transport inside. So that is the principle that we are going to use here. People have tried, that a tapered fiber, bent fiber, or people have tried with, nanoparticle coated fiber or antibodies coated fiber. Different modalities are there. Let's look at each of them. Before going to discuss about each modality, let's look at how these fibers are actually working. This is the basic principle, total internal reflection, which we already studied in 4th standard or 5th standard, the mirages concept. Why mirage is happening? Because the total internal reflection is happening from the rarer medium to the rarer medium. The light is actually reflected. That's why mirages are happening. The same concept people are actually using. We are also using here. The light is actually transported from the denser medium to the rarer medium. At a particular angle, it will be reflected to the same medium. So this is the total internal reflection, and the angle, call it as a critical angle, the critical angle. So you see that if the light is above the critical angle, when I say above the critical angle or greater than the critical angle, I'm talking about the with respect to the normal incidence. That normal is there at the dotted line. With respect to the dotted line, if the incident angle is greater than the critical angle, it will be guided inside the fiber. If it is less than, the light will be reflected, going out of the fiber. You see that there is a dot in the second figure. There is a dotted line which is out of the critical angle, which is going outside the fiber, and all other lights, for example, the black color and the red and green color lights are actually going inside the fiber, and they are all guided. They're all guided through the fiber, and this energy will be intact with the fiber, and it will be delivered at the other fiber tip. So while this light is going inside, there will be a electric field concentrations are happening. This is called modes of the light. Each each light ray that is actually transported here, you see that there is a different multiple times it has to reflect to and fro from the axis of the fiber, and you see that they are actually going to create some interference pattern. So these interference patterns are called modes here, and different modes are actually happening. The first mode is like Gaussian distribution, second one is the sinusoidal mode, and the other one is again the Gaussian distribution. So likewise, there are different modes are happening. So we are not going to discuss in detail about them. However, you see that when the modes are happening at the core cladding interface, the mode is not actually a zero. It is not going to the zero. There is some extension. The light is actually extended a little bit to the core cladding region, and this is called evanescent region, and this evanescent light is going to helpful for our sensing activities. If any small change is happening, if any small change is happening at the surface, then this evanescent wave will actually immediately recognize them, because there is a change in the evanescent field there. Either if it is absorbed or transmitted or disturbed, something will happen. So then you can get to see the mode will disappear there. So this is more important for the sensing. However, you see that there are different, the silica fibers are there, PMMA fibers are there, and step index, grid index, single mode fibers, multi-mode fibers. There are different fibers are also there. You can actually look into the details of them later. So now, actually the fibers are actually initially designed for the medical applications only, to actually see the tracking images. They wanted to keep a glass rod inside and wanted to see what is happening inside. After that, people actually tried, this is actually light transporting is good, so why don't we use for communication purposes? Of course, the fiber optics are developed for the low loss light signal propagation purposes, for communication use only. Later, people realize that why don't we use, if the light is transported and it is sensitive to its environment and its intrinsic properties, why can't we use this fiber optic as a sensor, including the different sensing phenomena, for example, fluorescence spectroscopy, or the absorption, luminescence, or Raman scattering, or SPR principles. There are different principles. In association with them, why don't we use these fiber sensors as for applications into biosensors or maybe any physical sensors? Yes, the people have tried it. So now you see that the advantages of the fiber, because of this miniaturized size and the remote and real-time monitoring capabilities, because you can actually, the single fiber, small fiber, you can keep it anywhere, you can send it to anywhere, any place, and now you can actually detect the molecular presence over there. So now you look at this applications of fiber optic. There are many applications, including lab on chip, lab on fiber, lab inside fiber, or lab around fiber. You take, there are many applications are there: medical diagnostics onwards, chemical sensing, temperature sensing, environmental monitoring, pressure sensors, food safety. There are different applications are there. Now we are going to discuss about biomolecular sensing in this class. So when I say fiber optic sensor, as of now we discussed about sensor. Now we are going to specifically talk about biosensors, how these fibers become biosensors. Let's see. Different modalities are there. For example, you see the fiber can be like anything. The fiber can be a tapered fiber, or tip fiber, or decladded fiber, or maybe a fiber graded fiber, or bend fiber. You can take any different modalities. Now there is a different concepts that we are going to do, whether it is a reflectance concept, transmittance concept, absorbance concept, or fluorescence, change in RI, or SPR, LSPR. There are different concepts that we can associate for sensing applications. However, when you add a bioreceptor, to the sensor, then it is become a biosensor. Here the bioreceptor added is the antibody molecule, which is a Y-shaped molecule. So now you see that the moment I add antibodies to here, this sensor become more sensitive to and specific to a particular antibody only. If you put any other molecule, it will not react. Only particular antigens can only sit onto the biosensor here. So this is sensitive because of the sensor that is you are using, this U-bend sensor, and it is specific because of the molecules that are actually sitting on your fiber. So there are so many people have actually done research extensively and given a lot of reviews and all. You can go through them. In my PhD, I used bent fiber because of its easy fabrication and ergonomic design, and I have found that something like fluorescence, chemiluminescence have some limitations. I used the nanoparticles instead of chemiluminescence, fluorescence. I used SPR based technologies here. So let's look at that. in detail how many types of fiber optic biosensors are there and how distinguished they are. We are going to discuss now. Now look at the fiber optic biosensors. The first one is based on operating principle, and second one is based on the recognizing element. So now when I say operating principle, again two modalities are there: intrinsic properties based and extrinsic properties based. When I say intrinsic properties, fiber itself, which means that fiber its own properties will get or changed. Extrinsic means fiber will have a helping molecule or helping layer or any other model where it is sensitive will be recognized by the fiber. So there are two things are there. So the first thing, intrinsic system will be having the RA sensitivity based and absorption sensitivity based. An extrinsic biosensor where SPR, LSPR, and MRR kind of concepts are going to be used. But if you come to the recognizing element, the recognizing element could be enzymatic based or affinity based, or could be nucleic acid. Affinity, when I say affinity, antibody based or nucleic acid based, could be anything. So let's look at the first thing, intrinsic fiber optic biosensors. People in that, again, I'm talking about refractive index sensitive biosensors only here. A lot of research is going on in this particular area. So most of the times people are actually looking for wavelength based analysis, which means a spectroscopic based analysis. Shift in wavelength will be recorded through the spectrometer. Then we are going to calibrate with different concentrations and all like that. That's what we're going to do here. In this specific wavelength based detection, grating types are available, interferometric types are available, and resonance types are available. When I say grating type, fiber gratings are there, long period fiber gratings are there. Interferometric means Moxander, Michelson interferometry. There are interferometric fibers are there, and the resonance means surface plasma resonance, localized surface plasma resonance, and the MRR, that ring resonators are also there. There are many modalities are there. Let's look at each of them. The second part is, first is the refractive index sensitive. The second part is evanescent wave sensitive. In this evanescent wave sensitive, at the core cladding region, there is a small energy that is leaking into the cladding region. It appears to be leaked into the cladding region, but it is intact with the core only. So this region is called evanescent field. If any molecule of interest having any absorption characteristics at particular wavelengths, so that will be absorbing a particular wavelength. When it is come closer to the within the evanescent region, a particular wavelength will be absorbed. So this phenomena is also being used for fiber sensing applications. I'm going to discuss this particular technology in next slides in my research topic. So let's look at the declouded fiber evanescent wave here. However, we are not talking about the evanescent wave absorbance. Now only talk about refractive index. What do you mean by refractive index? The molecule does not have any absorption characteristics. The presence of molecules will change refractive index of the medium around the fiber. In this case, people have tried the declouded fiber coated with antibodies, and another molecule, that antigen molecule they have given. So when the concentration is increasing, you see the binding kinetics are changed. However, here what is happening, the light which is transported inside the fiber, for example at 1550, you get to see there is a peak. That peak is actually shifting towards right side, towards the higher wavelength side, upon which the binding event is happening. What is happening here, when the molecule is coming at binding to the surface, what is happening here is the refractive index is changing. The change in refractive index, because of the changing in refractive index, this wavelength shift is observed. So here there is no absorption is happening, it is only simply a shift in wavelength is happening. In the second modality, people tried with the absorption. For example, if you have a molecule which has absorption characteristics, which eats the light. For example, methylene blue absorbs a particular light. If you use this particular molecules, the moment this molecule come in contact with the fiber, the particular light will be absorbed by this molecule, so that in your spectrometer, that light will disappear. That particular wavelength light will be disappearing. So how much amount of the molecule that are actually binding, that much amount of the light at particular wavelength will be reducing. So you can calibrate again and find out what could be the concentration here. People have tried with bovine serum, albumin, and the methylene blue. People tried with tapered fiber. This is some results are given here. And actually, people with some other researchers are actually tried with interferometric sensors. We talked about RA and evanescent wave sensors. Now it is interferometric sensors. what happened here is Moxander interferometry and Michelson interferometry. People have tried both. In these cases, you see that the antibody concentrations around 0.1 nanomolar and 4.59 nanomolar. People have tried with Moxander interferometry and Michelson interferometry. However, at this experiment, they found that Moxander interferometry is better than Michelson interferometry. So this is people have tried, and biomolecular detection has happened with this. And after that, some other fiber Bragg rating rating techniques are also used. What does it mean here? The fiber Bragg ratings are actually rejecting a particular wavelength, and this rejection of wavelength is sensitive to its environmental changes, which means that the rejection of wavelength are actually shifting here and there by changing its refractive index around the medium. So any molecule that comes and bind here, changing refractive index will happen, and the rejection wavelength, reflected wavelength will be shifting here and there. So identifying this wavelength shift as a graph, you can see that the different biomolecular sensing is also happened here. And the next one is decladded fiber, simple decladded fiber. People are trying with, why can't we use the absorption characteristics of molecules itself, like we said in the methylene blue, it is a chromophore, but now why don't we use its own absorption characteristics of the molecules? For example, if you take the globular molecules, they absorb 280 nanometers light. So then use the UV light and see what is the binding kinetics and all. People tried with that, and you see that the binding events are being captured for different concentration. The different binding events are happened, and people try with the lowest concentrations identification is possible with this technology. Next one is, why don't we try with the higher big molecules, for example, E. coli and all? Yes, of course, people tried with that. Even E. coli also having the absorption characteristics at 280, people tried with that. Of course, the straight fibers and Lubin fibers have been tried. You see that the binding kinetics have been given, and you can actually find out very low concentration, 10 to the power of 3 CFS people have actually tried. However, you see that there is one small problem in this technology is, any immune system molecule, for example, antibodies or maybe some biomolecules, they're all absorbing at 300 and 280 nanometers light around here and there. But how to identify them, whether the molecule is a particular molecule or a different molecule which is absorbing the same wavelength? So the specificity is being derived from the molecule that is being coated on your fiber, and of course, then people have tried with different other molecules are also given. The non-specific binding people have tried, they are actually not giving any response. After that, people have actually tried with extrinsic biosensor. We talked with the intrinsic properties of the biosensor, but when it comes to the extrinsic biosensor, the fiber is only the transporter of light. Fiber will not participate in sensing event. However, fiber is associated with the sensing element here, which could be a thin coating like a nanoparticle, like a thin film AU or TiO2 or silicon dioxide, another coating, or maybe nanoparticles can be used, or maybe ring resonators can be used, or any other external sensing element is to be attached to the fiber. Fiber will actually carry the light, and this external element which is attached to the fiber is sensitive to the environment, and that will influence the light that is flowing through the fiber. So surface plasma resonance and the micro resonator rings, people are actually doing it. Let's look at the one technology here. The fiber is being sliced, it is actually sliced slightly and coated with a gold film, and then other TiO2 film is actually given there. And now you see that the change in refractive index on the fiber, which is that on coating, if any change in refractive index, the absorption characteristics of that particular gold film is actually changing. So when I say the refractive index change from 1.32 to 1.35, the absorption characteristics of the gold is changing. That can be recognized by the fiber. So likewise, we can actually do that. When I say this technology, why don't I use coating on this gold nanofilm and then go for biosensing application? Yes, we can do that. People have tried that. So this is the fiber coated with a thin film and pass the light. Because of the gold presence, there is a particular absorption happening for the gold, and any environmental change around the gold will give you the change in refractive index and then change in the absorption characteristics of the gold. So now that can be recognized. People have tried with micro resonator rings and the sphere. People have tried with, and you see that these ring resonators or the sphere resonators are actually sensitive. These are the resonating at particular wavelength, and this resonant wavelength is sensitive to its environmental change. if any nanoparticle or any particle will actually come and bind to the micro ring resonator or this pair resonator, then you get to see the change or the slight change variation in the wavelength shift. And next, the importance of nanoparticles here. Why don't we actually use nanoparticle conjunction with the optical fibers? Yes, we can use, as we have already seen the Pcr technology where the thin film is coated. It is a two-dimensional structure. Why won't you go for the nanoparticles or the dots instead of a two-dimensional structure? Why don't you go for the three-dimensional structure, which is very small size in zero dimension, let's say, parts are the quantum dots. What is more important here is these particles, the plasmonic particles or the particles or silver nanoparticles, are actually different colors. It appears in different colors because of the size, its size. Now you see that each different particle, for example, a sphere or the rod or the long rod, you see that the absorption characteristics are different for different particles. However, these nanoparticles are very sensitive to its environmental changes. For example, if any other particle comes and touch the nanoparticle, and you see that absorption characteristics of the nanoparticle will actually slightly move. So this can be used for sensing applications by using the optical fibers. So people have tried with the lab on tape, lab around fiber, and there are multiple fiber technologies have come. Now this is another technology where the fiber is coated with nanoparticles, and these nanoparticles are sensitive to the environmental change. People try doing this and got very good sensitivities for the biosensor. However, the sensitivities are at different still not automolar here. It is in the nanomolar and micromolar only. So in order to improve the sensitivity, people tried with the U-bend structure here by enhancing the Evanescent field or enhancing the light interaction with the nanoparticle, and then they got very good sensitivity, at least 10 times better than the straight fiber. Here it is like, it is the intrinsic properties, or it is simply refractive index sensitive. It is changing only what we are doing. You are changing the refractive index medium around the nanoparticle. Then we are going to recognize the change in absorption characteristics of the nanoparticle here. But why don't we use the nanoparticle itself is a recognizing element? Before going to discuss about that, based on element, how many types of modalities are there? We can see the enzymatic reactions are also happening. HRP is coated. These Eliza based technologies have been done on the fiber, and people have reported this is very good example there, and people have reported very good sensitivities also here. However, people have tried with the fluorescence instead of enzymatic reactions. Why don't we go for fluorescence based technologies or chromophore tape based technology? People tried that, and here the immunosensing is happening and the nucleic acid sensing is also happening, and reported sensitivities almost like 50 micromoles and about 3.5 millimoles and all. The later fluorescence technology is being commercialized. The Raptor technology has come. It is available in market also. It is commercialized, and you see that the fluorescence being recognized by the optical fiber after the assay happened. Assay means that molecular binding is happening. And look at this, the other technology where people use the quantum dots. Why we use the quantum dots? Because after a particular concentration of fluorescence, it's because of its the high, after that the fluorescence quenching is happening. Instead of doing the quenching, we wanted to enhance the fluorescence value, fluorescence emission. People have actually tried with this quantum dots. What we do here, the quantum dots are actually absorbing the light from the fiber and then delivering that light intensity, that energy to the fluorophore molecules, and they will actually brightly appear. So this is the technology people used, and after that we got a small idea that why don't we use a nanoparticle as such as our recognizing molecule? Yes, we can use that. That is what I have worked. So from the previous literature, what I understood is U-bend fibers are much better than your tapered fiber, because tapered fibers is the handling is definitely difficult. The U-bend fibers are very easy to handle, so I have adopted the U-bend fibers because of its high sensitivity compared to your straight fibers and all. And I adapted the immunosensing technology where the ELISA is happening, not ELISA, that the nanoparticle technology is happening. LSPCR is happening on the fiber, and nanoparticles can be used as a recognizing elements. And I have reported in a very, very low concentration, that is 0.17 ziptomole, or like maybe 100 molecules in 25 microliters is possible to detect within 25 minutes of time. So this is what I have done in my PhD. And after this, what we understood is, of course, if the nanoparticle size can be varied, or or you can actually use different nanoparticles here. So I tried with different particle size, with different nanoparticle size. Then I have actually observed that there is a change in its dynamics. The sensitivity dynamics are there. So you can, if you use a small particle, you can actually recognize somewhere around nanogram per ml to microgram per ml. But if you wanted to use much more sensitive, go with a little bigger particle and again increase the particle size. Likewise, you can actually do 30 nanometer, 40 nanometer, and 40 nanometer plus silver enhancement. I did all these types of assays to enhance the sensitivities. This is very, very important research that we have actually carried out, and you see that the total advantage here is we are going to do analysis without a spectrometer. Here you don't need a spectrometer here because we are actually not looking at the spectral shift. We are only looking at the absorption, which means that only one wavelength, how much is the intensity variation. So it don't require a spectrometer, so that we can go with PD, which is actually reducing the cost of your diagnostic procedure, because you don't need a spectrometer, which is costly equipment, and only with a small LED and PD and a small fiber and a little amount of nanoparticle solution is itself is enough to carry out this entire work. And you see that that reduced sample volume, almost like only 25 microliters of sample is required, and minimal use of reagents, 25 microliters. Of course, after that we reduced it to 10 microliters also, but I'm not reporting here. So this time of your analysis is only 30 minutes. You give your sample, and after 30 minutes collect your report in a very precise manner. Please try to understand, when I say collect report, this is a very precise manner, very, very, very low concentrations you can identify. And after that, our colleague has tried with nucleic acid based assay with nanoparticles as just a minute. Nanoparticle being used as a recognizing element for DNA assays also. Instead of using the immunosensors, we can actually go for DNA assays also. We have reported almost like 25 microliters, like 5 automolar or 1 picogram per ml DNA can be recognized, or 0.2 picomole or 1 picogram per ml or 5 automoles in 25 microliters can be detected using this technology. What you require is a simple light source, which is a laser light source, and we need a photo detector. Light is actually traveling through the fiber, and this, when the nanoparticles comes and bind, and this hybridization happening, this hybridization, very specific hybridization, and nanoparticle presence will give you the number of hybridizations that are happening on the fiber, and you can actually calibrate with respect to concentration, known concentrations. So this technology is actually we tried in our lab. And the other one is the very important, where we adopted this technology. One of our colleague actually done this work for the tuberculosis analysis. For example, if you wanted to find out the TB, we have actually adopted the same sensing modality, U-bend fiber and the nanoparticles as your recognizing elements. What he has reported here is, you see that tuberculosis, almost like 1 femtogram per ml. If it is very low concentration, 1 femtogram per ml is also able to detect by your sensors. So you see that the tuberculosis molecule is actually, that is called LAM molecule, man-LAM molecule, MTB-LAM molecule. This is the molecule. So we have antibodies, specific antibodies are there. One is coated on the fiber, second one is coated on the nanoparticle, and this specific reaction is happening and tapping or trapping the MTB lamp, and then you see the number of nanoparticle that is sitting on the fiber will give you the concentration. So here 1 femtogram per ml we have identified for the TB. This is actually published recently in 2020 itself. So the next one, the other work is for chikungunya, people in IIT Madras and IIT Delhi. This is the current work they're actually doing it, and of course this actually came into the Analyst 2020. It is now published. Now you see that the detection limits are 0.5 to 0.52 nanogram per ml or 8.6 picomolar. Very, very low sensitive, and you see the linear range is 1 to 10 power 4 nanogram per ml, and the sensitivity is also given. This is for chikungunya. This is for chikungunya. The first one I showed you here is for the TB, tuberculosis. This is second one is the chikungunya, and the next one is for COVID. This is the same technology. The same technology we are going to use here. This is one of our colleagues from IIT Madras. So here also the same thing. We're going to use the same fiber and the light approach, where the light will be trapped by the nanoparticles when there is a COVID molecule or the COVID virus molecule is present over there. To see that this is how we are going to detect. Of course, this is again, it's in the research mode only, where people are try doing it. Maybe another 2 to 3 months it will come. There is a publication, expected publication from this. This is an idea, but people have been successful for tuberculosis and the chikungunya. Both people have tried and got the results, very good results. And people now in our laboratory, in my previous laboratory, people are working on COVID. The conclusion or the summary part is the biomolecular detection for conventional methods I have discussed. For example, ELISA, PCR, and what are the limitations over there I have discussed. And other state-of-art technologies also, Simoa, biobarcodes. These are the technologies I have discussed. And the next one is I've discussed about the fiber optic sensors. Various types are there. For example, you about the intrinsic and extrinsic fibers, how the biosensing is happening on that we have discussed, and the role of plasmonic particles are also discussed. And the next one is the current developments. What are the current developments in our laboratories are actually discussed. If anybody wanted to work in the fiber optic sensors with plasmonic particles, please feel free to approach us. However, currently we don't have facilities here. The facilities are in the IIT Madras. If you are interested, you can contact us. Then we can write a proposal for that work to the IIT Madras and get the laboratory access, and still we can work over there. However, this COVID time, maybe the COVID precautions need to be taken before we are going to the IIT Madras for work concerns and all. So thank you very much for this opportunity. Thank you very much to the E&I department just giving me this opportunity to talk about my work and the current developments in the biosensor field, fiber optic biosensors field. So any questions, please let me know. Please let me know any other questions from the audience side. Any questions from the audience side? Madam, Vimla ma'am.
Yes, sir. Participants, do you have any doubt?
Any query, they can actually contact me. Any query, they can actually contact me. Are you able to see the screen? Are you able to see the screen, ma'am?
Yes, sir. Yes, sir.
Yes. Okay. Any audience, anything that we want to discuss, please let me know. Anyways, I have given my mail ID is here, contact mail ID, but be at the rate of srmist.edu.in. You can contact me through mail so that I can respond to you. Any collaboration works, we will welcome you. No problem. You can write to us.
Sir, I have a question.
You tell me, sir.
Sir, actually, we always deal with the this characterization of so many materials. So, can we access any kind of data sets for the materials, particularly?
Data sets in the sense like for you're talking about nanomaterials also.
Yes, sir. Yes, sir. Like if I use lithium niobate as a piezoelectric transducer or piezoelectric material, and with that polyisobutylene type of material, if I use, then can we go for any kind of data set? Because I'm not finding out those data sets.
No, the data sets I am not sure, but you can talk to metallurgy people from IIT Madras. They can help you for the data sets and all. However, this fiber sensors can actually give you the spectral characteristics. If at all, if you're titanium or maybe the nibin, if you're using
Yes,
those,
sir.
if they are actually absorbing some particular wavelengths and all, you can actually look at the spectral characteristics of them. It is possible through fibers, and you just need a spectrometer in hand. But the data sets, I'm not sure. You may go through the literature only, but if anybody published some work on that, we can take that as a reference and then identify the particular peaks, and we can start working on that. But particularly material data sets for spectral analysis or the electrical characteristics, we need to talk to the metallurgy people from maybe from IIT Madras or any other universities also.
Okay.
The
Okay. Thank you,
expert,
sir. Thank you.
yeah, thank you very much. The audience, please feel free to write to me on my email here. That my email is available. So please, yes. Yeah, Mariamal, you're saying something in chat box. Just bring access to the chat box first. Okay, I just wanted to show you one of the recent work from our laboratory that is called, milk quality sensor. This is milk quality sensor done by our colleague. It was actually published. So if you are actually using a different milk quantities, for example, 3.5 and 4.5 and 6.5, the fat content, you wanted to know the fat content, then you can actually use this kind of sensors. So this is the recent work from our colleague. So if no questions are there, I'll stop the presentation.
Akshaya.
Yes, ma'am. Very good. I take this opportunity and extend a very hearty vote of thanks to the speaker, Dr.
Hey, your voice is breaking, Akshaya. The voice is breaking. So thank you, sir, for a very interesting lecture. I hope our participants would have a lot of information to take away from your
Detection,
work.
Biomolecule Detection, DNA Biobarcode, Fiber Optic Sensor. We are inspired by once again and
Aksha, actually, your voice is your voice is too much brave.
No signal.
yes, I'm sorry, sorry, sir, sorry, sir.
Yes. Yeah, it's okay. It's okay. Ma'am will continue. I think ma'am is saying something.
Yes, sir, it was very interesting, and I'm sure
Hello, sir.
to take lots of research ideas from your lecture, sir. Thanks for giving time for this lecture as well. In spite of the busy schedule and Pongal festival, you've given us this opportunity to listen to you, sir. Thank you very much.
Thank you very much, ma'am, for this opportunity. And feel free to contact me if you wanted to have some joint projects and all. Please write to us. We'll look into the possibilities from our side. Thank you very much.
Thank you, sir. Participants, please give your feedback. We will wait for another 20 minutes.
So can I, ma'am, or should I be?
You can sign out, sir. You can sign out.
Thank you, ma'am.
Sir, நாளைக்கு யாரு